Okay, so thank you very much. Welcome everyone to this distinguished lecture. I'm Xingu Yu. I'm the chair of RMIT Professorial Academy, hosting this event. So before we start, firstly, I really would like to acknowledge the people of the Kulin Nations on whose unceded lands we are meeting today. I respectively acknowledge their elders past and present. So today, what we are going to hear from distinguished Professor Magdalena Plebanski, a lecture on new frontier in vaccine and nanotechnology, which is, of course, is a topic of interest not only to research and medical science and engineering research, but also to much more to the public. All of us has interest, given our experience in the past few years. So, but before we start, let's just go through some housekeeping matters. This is actually a hybrid event, so we have people attending the lecture in person. We also have quite a number of people attending online as well. So for those people here, and you can just, as usual, just raise your hand after the lecture, ask your questions. For those people online, you can post your questions to the conversation sections. So at the end of the lecture, we will look at the popular questions and then we ask one by one to the presenters. Okay, so let's start the lecture by introducing the speaker, Distinguished Professor Magdalena Plebanski. She is internationally recognized award-winning researcher, and her focus is on understanding the immune systems in older individuals and development of practical immunotherapies and vaccine targeting cancer and infectious disease. In her role as enabling impact platform director at RMIT, she is promoting the establishment of multiple cross-disciplinary university-wide network, such as bioinformatics network, entrepreneurship club, mental health working group, and among many others. So without further ado, please join me to welcome Magdalena to deliver her lecture. So over to you, Magdalena.
Thank you, Xing, and thank you for establishing this lecture series, which allows us to really share some of our findings, but also share our passion for where the field is going in particular areas. I also want to acknowledge the people of the Woiwurrung and Boiwurrung language groups of the Eastern Kulin Nations on whose unceded land we conduct the business of the university, their ancestors, past, present, and emerging. And I also want to share some wisdom that I read from a book written by Uncle Paul Gordon and Paul Callahan called The Dreaming Path. In this beautiful book, they talk about how we all have different stories, but if we are following a dreaming path, we are united and heading in the same direction. So I think that is so relevant to research. We may all bring different aspects to particular strands of research, but if we're heading with the same dream in mind, we're all heading in the same direction. We're all together. So before I start, the first slide, in case people run off to do other things, is the acknowledgement slide. And I want to acknowledge both our group here at RMIT, the Cancer Research and Vaccines Research Group at the School of Health and Biomedical Science, College of STEM, but also our very many collaborators across all of Australia and globally across multiple institutes and our funders. And more than anything, I want to acknowledge the volunteers who participate in our human clinical trials, because they're giving not just their blood, they're giving their time, and they're showing their confidence that we will actually make good use of every single precious drop of blood. So, thank you. So, today, vaccination. Oh, by the way, I have divided the talk into 4 sections, and I'll just give a little summary at the end of each section. So hopefully that will wake you up at the end of each section. So, vaccination. We know this, right? It's effective at the population level, and it can both prevent disease as well as provide long-term protection. And in fact, you can see on the very right-hand side here, just taking it off an NIH site, how vaccines have all but eradicated a number of diseases globally. And this is something we often forget. We really, in certain countries around the world, we really don't have polio, diphtheria, we don't have measles. There may be pockets where some of these diseases, particularly measles, occasionally emerges, but we have all but eliminated major infectious diseases that used to kill hundreds of thousands of people. So it is an effective demonstrated strategy at the population level. Now, COVID-19. Most vaccines prior to COVID-19 were a little bit of the pathogen, that is the infectious agent, either attenuated, so the virus is or bacteria is inactivated, so it's no longer pathogenic, it can no longer cause disease, or bits and pieces from them mixed in with something called an adjuvant. An adjuvant is something that I'll talk a little bit more about it later, but generally something that just irritates your immune system, says there's something here you need to pay attention to. It calls attention to it. So before COVID-19, there was literally just one adjuvant license for large-scale global use, and that was alum. Now, what were some of the limitations of that? Well, pretty much a limited range of diseases that you could tackle, because you had to know the pathogen to attenuate it or to find the bit of it that actually would induce the appropriate immune response. The alum adjuvant tends to induce antibodies, and antibodies can give you one type of protection. So protection used to be limited by antibodies. And the way that the vaccine field had moved was incredibly slow. So literally taking, at the minimum, when I used to lecture, I used to find that it was a minimum of 6 years for even a small tweak to a new vaccine to come and be translated into use. And if you're talking about a brand new vaccine, it used to take conservatively 15 years from big, exciting laboratory results to actually being able to implement it. So the timeline used to be very slow. And it means that even responses to new strains of a virus, new strains of a bacteria, used to be incredibly slow as well. Now, also, and I'll talk a lot more about it, so I won't explain what it is now, but all these vaccines would not consider non-specific effects. Now, new opportunities since COVID-19 and the new type of vaccines that had come with it. The nanoparticle ones, and literally most of the vaccines that have come online in COVID are nanoparticles, one way or another. They are adaptable to diverse targets, diverse antigens, diverse pathogens. Some of them can also induce cell-mediated protection, and the immune system has two arms, antibody and cell-mediated, so now you're finally able to engage both arms. And they tend to have more agile, streamlined production, and therefore can respond in a more agile way than, say, 6 years to a new strain coming on. So, new frontiers, though. The talk is not about this, and I'm not, I'm not here to plug any vaccine. I'm here to talk about the new frontiers. What's next? What's after this? And the new frontiers that I'm particularly interested in are personalizing vaccines, making vaccines really consider the individual they are being given to, and harness beneficial non-specific effects. And you're not by yourself in this room saying, what do you mean beneficial non-specific effects? Even people who are deeply in the area of vaccination research don't know what beneficial non-specific effects are. So I'm going to tell you about it. So literally, it's like putting your immune system in a gym. And that workout for your immune system has to be right for the environment you live in to get the beneficial effect. So there's kind of no point exercising your arms if you live in an environment where you have to run away from a tiger, right? So similarly with your immune system, the way in which you gain this non-specific effect is by exercising the right arms of the immune system and legs for the environment you live in. So non-specific effects were initially discovered by epidemiologists when they were studying vaccines in Africa, particularly Professor Peter Abbey and Professor Christine Stabell-Bell from Denmark. And they found this amazing thing, which was that vaccinated babies in certain areas of Africa, and they primarily worked in Guinea-Bissau, when they were immunized, their mortality would dramatically drop. And this was not just from the disease they were vaccinated against, it was all-cause mortality. So literally from gastrointestinal, from lung disease, when you got an oral polio vaccine. So it was just like, what's going on here? And since then, there has been a lot of activity in this area, and it's been validated. The WHO has validated that non-specific beneficial effects exist, and that also said we don't understand them. We need to understand them. One way in which, this is just a bit epidemiology, but one way in which non-specific effects are actually being harnessed right now in the clinic is with some vaccines like the BCG, the tuberculosis vaccine. And the tuberculosis vaccine is being used at the moment in the clinic as a treatment for bladder cancer. So it's a bacteria vaccine used to treat a cancer. So you couldn't think of two pathogens that are more different. So what we found, however, is that this workout to the immune system differs depending on your biological sex and gender. And these are just some publications with our collaborators and ourselves where we've seen this pattern again and again. The early changes in all the genes going up or all the genes going down, expression of the genes in the immune system differs. Whether you're male here at the bottom, I think it's, oh no, female at the bottom, it's the DTP vaccine, just an example from a paper. And in green is transcripts of the DNA going up. In red, it's transcripts of the DNA going down. And these are females and these are males, 24 hours after the injection. So how a vaccine affects you will depend for the non-specific effects, whether you're male or female. In fact, generally, how your immune system reacts to vaccines, not only for these non-specific effects, but generally there will be differences between males and females, and this goes all the way through the lifespan, from babies to older adults. So the message here is that these non-specific effects of vaccines, the potential beneficial harnessing of them, has to also consider that they're sex and gender specific. Overall, females, particularly in the older age, tend to be more what we call inflammatory. So they tend to, for most vaccines, have higher immune responses, induce higher levels of antibodies. And at the same time, they tend to get more inflammation. And you can see that because of, say, you get a sore arm. But, oh, no, don't want to go forwards. What's going on? Can we get help? Or has... Yeah. Okay, so we all have better things to do, so I'll just keep talking, and then you'll see my pretty slides, right? So, so in older women, there's a particular phenomenon called inflammaging, and so it's a made-up word that now has made it into the scientific vocabulary. Ooh, great. Maybe the battery has fired. Okay, so you get more inflammation when you're older, inflammaging. But the problem is, inflammaging is actually associated with a lot of not so good outcomes for older individuals. Lots of mental capabilities, cognition, frailty in older individuals, which is again associated with accidents and oh, great, and healing that may be impaired. So, and this is not trivial for us. We are experiencing in many countries in the world, we're experiencing longer lifespans, which is a good thing. I'm all for it. But that does bring with it the fact that we have some populations where literally a third of the population will be over 65 by 2050. So a third of the population is now in this basket where the immune system may start having these inflammaging effects. Moreover, given that as we get older, our repair and control mechanisms for our cells go down in the body, and our immune system changes with age, we are fully expecting, and it's predicted, that there will be an about 50% increase in cancer, as it was estimated in 2010 to 2030. So we're going to have more cancer, and we're going to have an aging population and many of the diseases that are associated with older individuals. What happens to the immune system? It's so central. Now that little diagram there is for the scientists in the audience and online. But generally, what we are having with the immune system is that because of the way that the immune cells are generated from factories like the bone marrow and editing places like the thymus, less naive, inexperienced immune cells are generated. So you generate less possibility of seeing new things. So the immune system gets a narrow attention span, and it fails to recognize new dangers more and more. We also, as we get older, with changes on what the immune system recognizes, the immune system gets a little bit confused, and it tends to hyperreact or react to things it shouldn't, including our own self. So we all know that things like, say, rheumatoid arthritis is increased in older individuals, so autoimmune diseases. We also, and this goes back to the inflammaging point, the immune system also starts hyperreacting in the wrong ways, just producing cytokines that are actually too many of them and not quite right for whatever it is that the body is being confronted by. All of this means that in older individuals, there are massive implications for vaccines. And we have actually seen this, interestingly enough, during the COVID pandemic. So when individuals had their course of vaccination and then just analyzed month and months later, the 7-month cutoff was particularly interesting. And if you were under 60 years old, 29%, only 29% would experience what's called a breakthrough infection, i.e. you're vaccinated, but it was a very long time ago, you may still get infected. If you were over 60, that got up to 93% of individuals experiencing a breakthrough infection. So massive implications of your immune system changing with age as to how your immune system sustains memory, remembers stuff, and how it can manage a new infection or disease. So one of the studies that we're doing at the moment is called Vaccine Immunomodulation Throughout the Aging Lifespan. It's a phase 2 randomized clinical trials, and it's looking at flu vaccines and diphtheria tetanus vaccines in our beautiful volunteers in Tasmania. And the volunteers give us blood samples before and after vaccination. They also give us stool samples, and I'll come back to that in a minute, as well as we do questionnaires for cognition, mood, and various other aspects of their initial clinical profile. And then, what do we want to know? And I was just discussing previously with Zing that these days we have such an opportunity to really maximize the information we get from each one of these incredibly precious samples. So what we do are multi-omics analysis. What do we want to really achieve with this before I show you what analysis get done by us and others more and more globally? Well, we want to understand the immune system. We want to understand how it reacts and how it reacts to different vaccines. In this particular trial, we're trying to understand how it reacts to 2 very commonly used vaccines in older individuals. Flu, which is actually recommended and certain formulations given for free, and DTP, which is recommended. So we also collect the stools because we know that there's a massive interaction between the immune system and the gut microbiome, and it's a two-way street. So your state of inflammation may influence what bugs you have in your stomach, beneficial bugs, microbiome, and your microbiome will in turn act on your immune system. And more and more then there's additional consequences because the immune system interacts also with the brain. The immune system interacts if you have a disease, for example, cancer. So this triple axis of immune system, gut microbiome, and the brain is something that is becoming more and more intricately linked as we keep doing research, and we need to understand this in a cohesive way. So, multi-omics. What do we do with every precious bit of sample? Well, we analyze all the cells that are circulating in the blood, the immune cells. We analyze the microbiome. We analyze from the cells that are circulating in the blood. We're trying to see all the genes that have gone up and all the genes that have gone down, and we do it at a single cell level. So in diverse cells, different genes will come up and down, and we're looking at the cells individually, 100,000 cells, and we're looking at about 60,000 transcripts in each cell. So the amount of data that is generated on the inside is just extraordinary. Obviously, we're also looking at antigen-specific antibodies and epigenetics. Epigenetics is a very exciting one for us to look at because it tells us not just what's there, what has responded, but what is the potential of the DNA to respond to something. So it's not about a DNA sequence, it's about little bookmarks that we put as we grow older over our life. We put little bookmarks in our DNA this gene I'm using a lot. Let's put this bookmark here. This gene I really don't want to be using, so I'll put 2 tags there to switch it off. And more and more we're understanding what these tags do and how to find them, and we can map them. So it's not just about an individual responding to something. You can look at their DNA and say, this individual will tend to respond to a vaccine stimuli with production of high levels of TNF, which is highly inflammatory. And you can do all this without doing anything to the individual or even looking at the TNF itself. You just know that that person has that tendency to react in this way. So epigenetics. We're also looking at the potential influence of other infections, like cytomegalovirus, which is very common in our population. Literally all of us, not all of us, but let's say in anything between 50 and 80% of individuals will have cytomegalovirus just integrated into their bodies, and this will affect the immune response. Okay, and then doing cell assays on those donated cells in the lab. So I'm only going to give you one little result for fun, hot off the press, but I think maybe the question for us is more about, okay, so if you're older, should you get your vaccine? And there have been 3 vaccines given in Australia for flu. The standard flu vaccine in 2017 did not really work very well in older individuals. And the US at that point had also changed their formulations because said, no, actually older individuals have a different immune system. So they've changed it. And there's 2 other vaccines now. And Australia has followed suit. There's the high dose, which is basically the same vaccine, but just 4 times more of the vaccine in the vial. And the adjuvanted vaccine. So it's a standard dose, but you add something to wake up the immune system more in an older individuals. And now those 2, what they're calling augmented vaccines, are available for older individuals. So bottom line is, both the augmented and the high dose flu vaccines work better in older individuals than the standard low dose vaccine. So that's what my mom's getting. She's getting the high dose. But we were interested to see, well, if we were to think about harnessing those non-specific effects and start getting the immune system of older individuals to the gym, to get them working out aspects of their immune system to basically resist overall infections and other pathologies better. Is there still going to be a difference between old and young, between the different vaccines in terms of the non-specific effects they give? And would it be different in males and females? So hot off the press, we actually have looked at all the genes going up and down in our volunteers, and here you can see the standard dose, and in orange it's females and in green it's males. You can see, and blue is genes going down and red is genes going up. And you can see that there's a number of genes that clearly in males are going down and in females are going up and vice versa. So there's a different pattern for the standard vaccines. Same thing happens for the high dose vaccine again, but interestingly, the adjuvanted vaccine, there's a far less clear pattern of difference between males and females. What does this mean? We're still in the middle of this, analyzing it. So I'm just sharing an early preview, but bottom line is, in terms of non-specific effects, we may have to really consider which vaccine and if we're going to be giving it to males or females. So, conclusions from part 1. The non-specific effects of vaccines can affect multiple gene pathways and are age and sex dependent. And it gives us the, again, this is a frontiers talk, not what's there, but what do we see next? So what do we see next? Can we rationally engage these non-specific effects? Hey, do new things. Revitalize the immune system of older individuals by working it out in the right ways. Is this just a pipe dream? Or are we able to do a little bit of more in-depth research and start addressing some fundamental questions? So to get to the next section, I just need to give you a little bit of immunology. So this guy here, or girl, the dendritic cell, is the sentinel of the immune system. It eats things, takes them up as they come into the body, and then particularly particles. And if there's an adjuvant or some other danger signal present, these cells get very revved up to travel to the lymph node and tell the rest of the immune system what's going on and activate it. And in this way, we activate B cells and plasma cells to produce antibodies. And we also activate this other part of the immune system, the cellular part, and particularly cytotoxic T cells. And why is that interesting or important? Because we all know that antibodies will block whatever pathogen comes in from infecting a healthy cell. But once a healthy cell is infected and the virus, for example, has taken it over to make it a factory for itself to produce more and more virus cells, it's hiding from the antibodies. But the cytotoxic cells can see molecules on the surface of an infected cell and kill it. So we're basically stopping the virus from being able to expand and replicate in the body. So can we do this rational thinking about vaccine creation? And number one, when considering vaccines and advances in nanomaterials, is safety, 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 and I can keep going on. That's number one. The rest, for us, I would want to make sure that it is efficacious not just in one group, but we actually identify materials that will be efficacious in different individuals that have different immune systems. I gave you the example of old and young, but there's other groups. For example, pregnant women have a very different immune system, and other considerations, safety, 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 and safety, that we need to take into account. Also, and this is a frontier, we would want to engage these exciting new non-specific effects. So for some time now, we have been interested in just going very systematically and rationally about vaccine design. So most vaccines that have appeared and are in use actually are pretty random in how they initially were used, because historically they were used, they inactivated the pathogens themselves, they were seen to be safe in a large population, and they kept being used. But perhaps we have an opportunity to just think it through. So, fundamental properties of nanoparticles. Nanoparticles are between 1 and 100 nanometers. And just to put it in scale for you, and I know that there's some nanotechnologies here in the audience, but I will do this anyway. So the difference between a 1 nanometer width, and it's 100,000 times smaller than the width of a hair. Now, if you think about the width of a hair, it's 100,000 times smaller than the width of a house. So that's the scale. And we were wondering, well, what can we start systematically investigating? And the first thing was size. And strangely enough, at the time we were doing that, that question had not been asked. What is the right size for a vaccine? We were particularly interested to see that dendritic cell, that sentinel of the immune system, did it have any particular preference for gobbling up any particular particle size? And when we gave the dendritic cells different particle sizes, lo and behold, we got a surprise. it does have a preference. It doesn't consider all particle sizes the same. Between 40 and 100 nanometers is the optimal size. So there actually is an optimal size for a vaccine. And then when we were able to alone or collaboratively conjugate different things that we wanted the immune system to see for a vaccine, bits of protein, bits of peptide, what we found is that basically we could induce massive immune responses, including both those CD8 T cells and high level of antibodies, without doing anything else. We didn't have to add an adjuvant. We didn't have to do anything, as long as we got that specific size right. And the type of immune responses that we were able to induce are the type of responses that could tackle pathogens that still need vaccines. For example, malaria with antibodies and Th1 T cells, or cancer where CD8 T cells play a major role. And we went down a pathway in terms of seeing and confirming that this principle will induce good immune responses in larger animals, as well as is capable of being scaled up. So, size. The proof of principle particle that we were using at the time, comparing all the different sizes, then we thought, okay, but what if we use the same size but use different materials? Now, not all materials worked, but we did find a whole range of materials where if we got the right size, this particular example is just an iron oxide core surrounded by a sugar. And we were able to confirm that they're non-toxic, non-inflammatory, and they're taken up preferentially by the dendritic cells. Then we did induce immune responses that were comparable to our gold standard particles. So these are the iron oxide core ones. These are our gold standard ones. And in fact, comparable to an adjuvant that is still experimental, but it's one of the most powerful out there. So yes, the principles do translate across different materials. So what about shape? Let's get more ambitious. So this is a very recent collaborative paper with the collaborators in Queensland, and bottom line is shape matters as well. And we found that these little guys, the rods, were the ones that were inducing the best responses, and this was against a bacterial infection. So not everybody can make particles in that tiny little size. So what about larger particles? And in fact, there's a whole literature out there, and people are busily and happily making vaccines that are larger size, and they still work. So why? And we believe that that is because a lot of these approaches don't just use the pure particle, they use the pure particle and something that will target your vaccine to that little guy there, the dendritic cell. So for example, receptors on the surface of dendritic cells, like danger signal receptors. And what happens when a particle gets taken up by this receptor, it also puts molecules on its surface that makes the dendritic cell more active to interact with the rest of the immune system. So that's an alternate way of getting to the same place. And in fact, we've played a little bit with that, even with magnets. And when we had our iron oxide core particles, we can also put them in places where we can artificially attract the vaccine to the injection site or to the in the culture to the dendritic cells. So dendritic cell targeting can be an alternate for larger particles. But then that's okay. But then we have to put these vaccines in the body. And what happens in the body? Well, what happens in the body is that all of our fluids, including blood and lymph, have a lot of proteins floating in them and a lot of antibodies. So what happens, therefore, is that particles get surrounded by a corona of our own proteins, which can include our own antibodies. And that is particularly relevant for nanomedicines that use a compound called PEG, and that includes some of the current mRNA vaccines, because there is limited data on repeat administration, and PEG is found in a lot of products. Cosmetics, your shampoo will probably have PEG. Medical contrasts for medical imaging, drug stabilizers. So question is, are we upregulating when there is an injection with a vaccine? Are we also upregulating the response to PEG as well as whatever the vaccine is carrying? So in this work, collaborative work and led by a DECRA fellow in the team, David Yu, we found that yes, absolutely, there is upregulation of anti-PEG antibodies upon injection with either the Moderna or the Pfizer vaccines. And when we then see whether coating the vaccines themselves in the sera from immunized individuals would change the way they interact with the immune system, they did. So we have more internalization, and this is just one example by a cell called a monocyte. It eats it more, internalizes it. So do we know what are the consequences of this? Not really. It could go either way. It could be that the vaccines, the more you administer them, the antibodies will actually clear them before they have a chance to induce an immune response against what you want them to induce an immune response against. Or it could be the opposite. It could be that because they're coated by antibodies, they're internalized by cell types that may provoke an even higher immune response. We don't know at the moment. But what we do know is that it's important to ask fundamental questions like this. And in fact, in another collaboration, we have collaborated with a group which has shown some alternatives that are non-PEGylated. And it's possible to make plexes with RNA that don't contain PEG. And at the moment, we're starting other areas of collaboration with additional groups to go down that path and found alternatives to PEG. So, but in the meantime, Moderna, Pfizer, Novavax, they're all being injected in large-scale human trials. So we want to understand what does happen with repeated immunization with the different vaccines. Does it matter what you were immunized in your first round to what then you get boosted with? Does it matter for your antigen-specific response? Does it matter for your antibodies? Does it matter for your T cells? So we're involved in large-scale human trials across all of Australia with our collaborators in PICOBA 1 and PICOBA 2 to basically start addressing these questions. So, conclusions part 2. Nanoparticle interactions with the immune system hugely dependent on fine differences in size and shape. Fundamental principles can translate across nanomaterials and species, and interactions can be modified by protein corona or by adding dendritic cell targeting ligands. I need to know how I'm doing for time. Okay, all right. So the next one, even more controversial, if that can be, which is, can nanoparticles have beneficial non-specific effects? So vaccines can have beneficial non-specific effects. Could nanoparticles have them? And therefore, all the mapping that we've been doing of immune potency, could it also be harnessed for non-specific working out of the immune system? So this is very controversial because the literature on nanoparticles very much comes from the field of pollution, and very much comes from the fact that ultrafine particles, and these are nanoparticles, so they're less than 100 nanometers, are the ones that are clearly epidemiologically associated with increased cardiac and lung pathologies. So no doubt about it, nanoparticle inhalation of pollution is bad. But when you put the microscope on this, these effects are affected by size, shape, charge, chemistry, and majorly by the adsorbed contaminants on these tiny particles. So can we look deeper? And we just wanted to find a material that was as neutral as possible to start doing our research. And strangely enough, we found polystyrene, which had been demonstrated to be inert and non-toxic in mice, sheep, and rats. And in our own studies in vitro on that dendritic cell, the DC, we found that these materials did not induce inflammation or reactive oxygen species, which is another bad thing to induce. So we were very excited to see this result, where basically giving the nanoparticles in the lung, polystyrene nanoparticles in a particular size range in the lung, didn't cause any inflammation or anything else, but also when then there was a challenge with an environmental allergen, basically this pre-administration prevented the elicitation of an allergic response in the lung. But at the same time, it did not affect the ability of the lung to clear an influenza infection. So here we have a very non-specific effect. 1 month earlier, you give a workout to your immune system in the lung, and then a month later, that lung, which normally would have responded with allergic inflammation response, doesn't do that. And if instead you challenge it with an influenza virus, it clears it, and if anything, clears it a little bit better than if they hadn't received the particles. So in this particular context, absolutely, it is possible to work out your immune system also in organs like the lung. So nanoparticles, physicochemical characteristics can be mapped to optimize immune responses, and nanoparticles by themselves could potentially be explored in this way to provide these broad non-specific effects. So old age does not need to be scary. It can be a little bit fun. So I do have another 4 slides. Yeah, is that okay? All right. So coming back to this, we were talking about personalized vaccine, old, young, male, female, and harnessing beneficial non-specific effects in vaccines and in nanotechnology as new frontiers. So this is, I'm telling you about things that are happening right now. It's not even an opportunity. It's a little bit beyond that. But end of the day, why are we doing all this? And these are just a few slides to tell us something that we are very committed about at the Cancer Aging and Vaccines Research Group, which is ovarian cancer. Now, ovarian cancer is the 7th cause of death of cancer in women. The problem is most women present at an advanced stage of presentation, and then there's very little that one can do. The late detection survival is only 5%, and the majority of women get detected at that stage. So we also know that the immune system is absolutely critical to control the tumor. And the tumor cell infiltration, which immune cells infiltrate the tumor, will tell you about the survival. But we also know most of our patients are older women, and at this point, we have, like we discussed before, a myopic, very narrow focus of attention, adaptive immune system, and a very inflammatory, hyperreactive, innate immune system. So in ovarian cancer, if we're going to be doing anything to engage the immune system productively, we also need to understand the immune system of older individuals. So now you can see why the lab is called Cancer Aging and Vaccines. And briefly, what we do is we engage big data analysis, like that slide I show you about big data and different sources of big data for early detection and for personalized therapy. We're collaborating with engineering, and Cesar's sitting there in the back, Cesar and Arnand, for not only finding biomarkers, which we do ourselves, but also really think about the translational process down the line to make this accessible to women to be able to test themselves or to be able to test themselves in the clinic. And these, we are very excited that we found some biomarkers that do enable earlier detection. And we also have found some biomarkers that enable us to personalize therapy. So from a drop of blood, we are coming to the point where we can tell you whether this drug or this drug is the one that you should take that will give you the maximum benefit for survival. So we're talking about drugs that would only work in 30% of women. Now we can tell which 30%, and they can take the correct drug. This kind of practicality, I'm just pointing out one paper there. It's even for women that have cancer right now, that are advanced stage. You can imagine somebody being in the middle of nowhere, in the middle of Australia, in the Antarctica. We have actually found that some biomarkers in blood can substitute for the initial alarm bell for things like an MRI scan in terms of sensitivity and specificity. So the practicality is always in our minds as well to provide benefit to women. The other part of this journey in terms of engaging anything we can really to help women with ovarian cancer is to collaborate in innovative new treatments. And we are collaborating with School of Science and another distinguished professor, Professor Bhargava, on some gold compounds, as well as the Hudson Institute and other, and obviously the Walter and Eliza Hall for new drugs and new immunotherapies. This collaboration with WeHi is really very exciting because we are able to engage multiple immunotherapies. And it's double blind, so we don't have the results as yet. But we're really going in depth with the big data to try to understand why some women respond to the immunotherapy and some women don't. And we're analyzing the immune system to an unprecedented depth, as well as with WeHi and Claire Scott's lab, analyzing the tumor tissue itself to an unprecedented depth. So we will gain enormous insight regardless of the results. And one of the insights that we are already getting from these results is to find new targets for vaccines in these big data, large-scale human trials. And you know, we have new targets. And as I discussed with you before, we actually have a commitment to develop new nanotechnologies. So if we put these two together, our aim really across our projects is to develop effective cancer vaccines, which basically means all of this that we've worked so hard to create becomes irrelevant, because hopefully this person will just not get cancer. So, thank you.
Have a few minutes. We have a few minutes for questions. Any question from the floor?
Thanks, Magdalena, for the very inspiring and interesting talk. I'll have a few questions. I'll just ask one question. The first part on how the immune system is age dependent, I wonder if you step back even, there's a whole aging question there, right? Do you know what happens to the immune system and why, the biochemistry of it? And then I think you discussed it a little bit at the end, can you reverse that? And it's an aging question.
Oh, 100%. Yeah, we're getting close to understanding the regulators of aging. And in the words of Indiana Jones, it's not the years, it's the mileage. And your immune system, how it's worked out. So the experiences that your immune system undergoes are as important as when that happens. So there's cumulative experience. And I mentioned the field of epigenetics. We are starting to see that particular epigenetic profiles are associated with better aging, and particular epigenetic profiles are associated with a better functioning immune system into old age. And the two tend to go together. So because, again, the immune system, cells traveling all over your body all the time, checking everything out, involved in repair mechanisms, they are so fundamental. If you can affect the immune system, there's definitely possibilities for helping age better. There was a second question there, sorry. Or...
Thank you.
Thank you so much. Great talk. Just one simple question about the non-specific effects, beneficial NSEs. So you shed some light on NSEs caused by nanoparticles about from the pollution, but can the mRNA LNPs containing vaccine cause NSEs?
That's something we need to look at.
Okay.
They don't know. If people don't know at the moment, and then that's why I say it's a new frontier. And this is something that the WHO has acknowledged that NSEs exist and beneficial NSEs can exist, but in terms of how many people are looking into that, it's still a frontier.
Good to know. Thank you.
Okay. Can afford one questions? I actually have a bit of question. I'm still a little bit later on this. Then I mean, it's fascinating to you talk about the shape and the size of the molecules and the nanoparticles. You can see the positive side. On the other hand, you introduce alien type of material, intersect with your cells. And what kind of procedures you have in place to ensure that kind of safety?
Oh, absolutely. And look, there's a whole slew of tests that you have to do for safety that are separate. And yet there's formal acute toxicity, chronic toxicity, and there's actually even, if it goes that way, there's actually companies that do that. So, the researchers, it's hands off. There's, you test it and you tell me. So there's a whole process to do that. In the lab, we have our own kind of shortcut version of things so that then we know what to focus our attention on to further progress. Inflammation, reactive oxygen species are very core, and more these days it's about this big data approaches as well.
Thank you very much. We have just 1 minute left. Anybody has any burning desire for the last short question? So if there's none, please join me to thank Magdalena for the fascinating talk. Thank you very much. And looking forward to see you in another distinguished lecture. Thank you very much. Thank you.
Thank you.